Okay, the lab number 3 of BIO206, spectrophotometric analysis of nucleic acids and proteins. Well, you should get actually two unknown samples, one DNA, one protein, and using the spectrophotometers determine concentration and other stuff. But first you have to dilute the samples because the spectrophotometric analysis always have the range of the concentration. But the stock solution is too concentrated that you would get good analysis by spectrophotometer. But DNA sample you have to dilute 10 times, and this which dilutes 10 times, dilute 2 times or 2 folds, and third dilution, which was diluted 2-fold, 5-fold. Now, when you have to dilute 2-fold, that means one part is your sample, one part is a solvent, solute, or water, buffer, whatever you're using. 2-fold. 3-fold means one part of your sample, 2 parts of water, buffer, 1 plus 2 is 3-fold. 5-fold, it's 1 part and 4 parts of the solvent, 1 plus 4 is 5. And 10-fold, it's 1 plus 9, 1 part of your sample, 9 parts of the water, buffer, 1 plus 9 is 10. This is very simple. The bigger problem for some students is that when you are defined with the total volume. The total volume of the first dilution of DNA is 200 microliters. But if you have 200 microliters and you have to dilute your sample 10-fold, well, divide 200 microliters by 10, you get 20. One volume is 20 microliters, that you add 20 microliters of your stock DNA to 180 microliters of water. The same situation, 2-fold, 200 microliters total volume, that means 200 divided by 2, it's 100 of the sample, 100 of the water. Last dilution is 5-fold, but total volume is 500 microliters. 500 divided by 5 is 100, that means 100 microliters of your sample, 400 microliters of water, buffer, whatever solvent you're using. But first, we use water for dilution. In the molecular biology labs, we use the millicube water. It's high purity water, and it's very expensive, but we don't pipette from this container. We have to first liquid water to the tube, which is labeled H2O, right? And lose the cap, pick up the pipette, blue, 1000. In this case, 1000 means one digit is red, and black 2, it's 100. Probably this was the beginning when they started manufacturing pipettes. 100 means 1000. Press to first stop using the pinky finger because it's a bigger container. With 3 fingers, pick up the water. I press already to first stop. Go to solution, pick up 1000 microliters, close, pick up the tube, open, and expel it with control. First stop, second stop, still pushing. I get out of the tube and I release. I have 1 ml of millicube water. Dispose the tip. Right? Now, we have 3 dilutions of DNA. I have DNA in one rack, protein in another rack. We need the DNA is 10 times, that you label the tubes on the frosted area 10 times. Another one is 2-fold. 2-fold, and the last one is 5-fold. 5-fold. Now proteins. Protein is 5-fold first dilution. Second dilution is 2-fold. And the last dilution is 10-fold. Okay, 10-fold. First we add water to the all tubes. With DNA, 10-fold, 200 microliters final volume, that means 200 divided by 10, 20, that we need to put 180 microliters of water to the first tube. 180, I have set up, pick up the tip, pick up the water, open, press to first stop, pick up 180, put it back, put to the tube. Okay, now I put the tube which I pipette into one spot further, that I have 2 empty spots between added, non-added. Second dilution is 200 microliters, it's 2-fold, that means 100 and 100, right? And I have 100, water, press outside of the tube to first stop, go to solution, and to water in this case, pick up the tube which you pipette into, go to the bottom, expel it, second stop, get out of the tube, release, close the tube. Again, I added here 2 empty spots, I'm going to add here. The third dilution of DNA is 5-fold, total volume 500. 500 divided by 5, it's 100, with 400 microliters of water for this dilution, right? That unlock the pipette, set up for 400, the blue, we'll go with the blue, water, I always, when I pipette, I always see the tip of the pipette, but I'm holding the tube this way, that I see what I'm doing. And the third dilution, expel it slowly, first stop, second stop, get out the solution, put it back, expel it. The tip. This is the water for DNA. Let's do the DNA dilutions, and after we go to the protein dilutions. But DNA, it's actually double strand, not high molecular weight, that you can vortex to mix or use other method to mix. I will show you. But first dilution 10-fold, 220 microliters of our sample of DNA. I set up for 20 and pick up the tip, pick up the DNA, press to first stop outside of the tube, pick up the solution, put the tube. Now I go back to the original setting. I have 2 spots at the beginning. I'm adding the DNA. First stop, second stop, get out of solution, release the push button, close, and I put again 2 empty spots between added, non-added. Now, pipetting by vortex, mixing by vortexing, that I just vortex DNA. I can do this, but I mustn't do this with the protein solution because it will form foam, and foam is denatured protein. But with DNA, another method of the mixing, gentle mixing, is you hold the tube like this in 2 fingers, and using this side of the finger, you just flip it like this. You see that it's mixing nicely. But this is DNA. With another method of mixing is especially for protein solutions, especially for protein solutions with catalytic activity, that it's pipetting up and down. But in this case, DNA, we have 200 microliters of the total volume, that to pipette up and down, to mix, you set up the pipette less than total volume. Well, let's say in this case 180 is fine. Pick up the tip, pick up the tube, press to first stop, go to the solution, and pick up to the first stop and expel it to first stop. Pick up to first stop and expel it. Pick up and expel it. Pick up and expel it to first stop, get out of the solution, second stop, and it's done. You have to do this several times, but it's a very good method for pipetting. But this sample is done. Now, second dilution of DNA, 2-fold and 200 microliters, that I have to pick up 100 microliters of the first dilution, because it's 200, 2-fold, 200 divided by 2 is 100, but one volume is 100. I set up the pipette for 100, pick up the first dilution, 100 microliters, and I have 100 microliters of water here, and expel it, first stop, second stop, still pressing to second stop, and dispose. And the DNA we can vortex, it's done. Third dilution is 500, 5-fold, that 500 divided by 5 is 100 microliters to 400 microliters of, in this case, water. That I have 2-fold dilution, pick up 100, okay, and this is our final dilution of DNA. DNA I can vortex, nothing will happen. This is what we do. Now, let's move it away. Now the protein. Protein solutions is 5-fold, 2-fold, and 10-fold. The first one, 5-fold, is, and the volume is 100 microliters. That 100 divided by 5 is 20 microliters. That and 80 microliters of water. But let's start putting water to tubes. It's 100, 5-fold, that's 80 microliters of water. I unlock the pipette, set up for 80, pick up the tip, water, pick up the tube which you pipette into, open, go to the first stop, second stop, and this is water. 5-fold, 100 microliters. Second dilution is 2-fold, and total volume is 200. 200 divided by 2, 100 microliters of your sample, and 100 microliters of water. That we put in water. Okay. First stop, get into solution, pick up, put it back, pick up the second dilution tube, 100 microliters of water. Okay? And again, in this case, one empty spot between added, non-added. And the third dilution is tenfold, and total volume 200, that it's 200 divided by 10 is 20, that one volume is 20, and we need to add 180 microliters of water. It's set up for 180. Okay, first stop, pick up the water. Oh, I don't have enough water. But, yeah, in some cases when you don't have enough, just expel everything, put the tip to the container, disposal container, and pick up the, don't hold like this or even like this with the solution in the tip. That I need more water, that lose the cap, well, 400 is enough, pick up the tip, with the pinky finger, pick up the cap, with these 3 fingers, pick up the container, press to first stop, go to the solution, pick up the solution, put it back, pick up, and add 400, in this case, microliters of water, which it's more than I need. Close the cap, pipe it down. Now, the third dilution of protein, tenfold, we need total volume 200, that means one volume is 20, 180 microliters of water. Yeah, I have 180, pick up the tip, pick up the water, press outside of the tube to first stop, put into solution, pick up 180 microliters of water, add to the tube. Okay, we have water, we go back one position with the tube. Sometimes when I was doing 50 or 60 tubes, adding several things, always after adding the one reagent, I moved back, because it's very easy if you have a lot of tubes to make mistake. Now, first dilution of protein, 5-fold, total volume 100, that means 20 microliters of our BSA solution to 80 microliters of water. 20, red pipette, press to first stop, go into solution. With the protein solutions, especially concentrated one, it's tricky. You just put as little as possible tip into solution, because if you put too deep, the outside of the tip is a film of the substance, especially proteins, which are sticky, that you're adding. If you put the tip to your water for dilution, you're adding more than 20, because outside you have 1 or 2 microliters. That as little as possible, and we put first stop, second stop, okay, close. Now we cannot vortex this. Perhaps, yeah, when I do the dilutions, I will vortex this first dilution. You will see how much foam it will be in the tube. Now we have to mix by pipetting. Total volume is 100 microliters, that I pick up 100 and set up the volume less than total volume in the tube, but let's say 80 microliters, and open, press to first stop, and pipette up and down to the first stop only, not to second stop. You do this several times, no bubbles. Get out of the solution, press to second stop, close the tube. This is our first dilution of protein mixed. Now, second dilution is twofold, that and it's 200 microliters total volume. That, actually this is tricky, even I forget to do this. Actually, we have two-fold total volume 200, but I put 100 microliters here to do that second dilution. But actually, I shouldn't do this, because here I have 100 microliters, and if I want to pick up 100 microliters when I have 100 microliters, I will never pick up 100 microliters because of the film outside or inside of the tube. We are unable to do this. But we should throw away this and label here two-fold, cross the five-fold, and add 100 microliters of water directly to the first dilution. That it will be total volume 200, and okay, 100 that I'm adding. First stop, second stop. I have 200, but the pipette is for 100, that I can pipe it up and down using 100 microliters. It's nicely, yeah. But if you have 10 microliters in the tube, you cannot pick up 10 microliters. You will pick up 9.5, something like this, but never 10. Because something will stay on the walls of the tube. But this is the, we have second dilution here, which is two-fold of the five-fold dilution, but 2 times 5, actually this combined dilution is 10-fold. That I will now, and this one we have to dilute 10-fold, and total volume 200 microliters, that I put, yeah, I put water here, 200 microliters, 10-fold, that I have to pick up 20 microliters of this and put to 100 microliters of the water, that it will be 10-fold dilution. Okay, 20. 200, yeah. That I pick up 20. Okay, dilution. First stop, second stop, and this. Now we have 200 microliters. We have to mix it up. That 180 is good. And yeah, pipetting up and down, only to first stop, second stop, still pressing. This is final dilution. But if you have 3 or 4 or 5 dilutions, doesn't matter, each different one, the final dilution you multiply 5 times 2 is 10, 10 times 10, that total dilution is 100 in this case, and DNA, it's 10 times 2, it's 20 times 5, exactly 100 the same. But we have our dilutions ready to go for the spectrophotometric analysis of the protein and DNA. Now we go to the spectrophotometers.